good morning or afternoon, depending on where you're joining us today. My name is Bailey Bostelman, and I'm the Director of Chapter Engagement here at the Foundation Fighting Blindness. We're so excited to have you here with us virtually today, and thank you for sharing your time with us. I would like to briefly review some logistical details for the call before we get started. Please be aware the controls are at the bottom of the Zoom interface. This control bar may collapse when not in use. If you prefer to prevent the controls from auto-hiding, you can use the following keyboard shortcuts to toggle the always show meeting control options. If you're using Windows commands, press the Alt key. If you're using a Mac keyboard, press Command and / at the same time. So we would like to go ahead and get everything kicked off today. I would like to first start out by welcoming Adrienne Keeve, who is our chapter president for our Jacksonville chapter. And she's going to share with us a little bit today and then get us kicked off. So Adrienne?
Hi, thank you, Bailey. So hello, everyone, and welcome to this webinar on emerging therapies and research for inherited retinal diseases. I'm Adrienne Keeve, and I am the president of the Foundation Fighting Blindness Jacksonville, Florida chapter. And I am excited to share with you today what's happening in our small but mighty Jacksonville chapter. I first came to the foundation in 2008, shortly after moving to Florida, because I wanted to meet others like me with Stargardt disease. Funny enough, I did not find anyone at the chapter who had Stargardt disease, but that was okay because I found plenty of wonderful people that understood the sight loss journey. So the Jacksonville area chapter was founded in 2008, and in 2009, we had our first vision walk. Since then, we've raised over $1 million for sight-saving research. The Jacksonville chapter has accomplished a lot in the 16 years that it's been in existence, including recently receiving the inaugural Luly's Light Award Guiding Vision Award. We received that earlier this year at the Visions 2022 conference in Orlando. This award recognizes the Jacksonville chapter's leadership in accelerating the foundation's mission by identifying stakeholders and community initiatives that can help increase revenue through events such as our annual Black Ties and Blindfolds Gala, our new See Like Mav golf tournament, and pursuing opportunities for matching gifts through partners such as Spirit of Exit's Dollar for Dollar matching program with Exit Inspired Real Estate. The Jacksonville chapter's success is largely due to the passionate and enthusiastic chapter leadership team. It consists of Dr. Emily Townsend Cobb, Dr. Sandeep Grover, Susie Ford, Lisa Pleasance, and Meredith Sauter. And I would like to thank each and every one of them for their dedication and commitment to the mission. I mentioned our Black Ties and Blindfolds event, and this is an annual gala with a dining in the dark component. It includes a silent auction as well. And the 10th annual Black Ties and Blindfolds just took place last month and raised over $15,000 to benefit the Foundation Fighting Blindness. So this event is hosted by our local 501c3 nonprofit Moms for Sight that was founded by our two past chapter presidents, Lisa Pleasance and Susie Ford, to help provide support for our fundraising efforts. Moms for Sight has raised over $115,000 since its inception in 2012, most of which has gone to our Jacksonville Vision Walk. We're gearing up for our 15th annual Vision Walk, taking place April 29th, 2023 here in Jacksonville, with our kickoff happening March 5th. Our Vision Walk committee is hard at work, planning an exciting day of celebration and community. Our walk committee consists of Susie Ford, Jenna Rollerson, Dr. Sandeep Grover, Meredith Sauter, Sue Townsend, Kristen Kirk, and myself. And I would like to thank each and every one of them for doing the best that you can to make this Vision Walk the best one yet. We also have a coffee social coming up in January, and we have the second annual See Like Mav golf tournament coming up March 4th. And you can visit momsforsight.org to learn more about that. We have a series meeting, hopefully taking place in Gainesville. We are technically the Jacksonville area chapter, so we go all the way up to South Georgia, down to Gainesville, and west to Lake City. So we have had chapter events in Jacksonville, St. Augustine, and Gainesville to try and extend our reach and share information about the foundation as far as we can. So if you're interested in learning more about the Jacksonville chapter or any other chapter, please reach out to the foundation staff and they can direct you to your nearest chapter. So today we have several wonderful presenters for you for this Emerging Therapies and Research for Inherited Retinal Diseases webinar. And you will hear from them shortly. And for more on that, I will hand it back to you, Bailey.
Great. Thanks so much, Adrienne. So yes, like Adrienne said, we have a great list of panelists here for us today. But to get us kicked off, I'm going to send it over to our very own Ben Shaberman. So Ben Shaberman is the Vice President of Scientific Outreach here at the Foundation Fighting Blindness. And he is one of our science gurus in terms of our webinar. So Ben, go ahead and get us taken away with our presenters.
Great. Thanks, Bailey. And thanks, Adrienne, for that excellent introduction. Congratulations to you and the Jacksonville chapter for all the great work you're doing down there. And welcome everyone else from around the world who've joined us for this webinar. We're delighted you could be here for what will certainly be jam-packed with all kinds of exciting research featuring presentations on emerging therapies from 3 companies, Nacuity, Opus Genetics, and OkuloVision. And each of our presenters will be presenting for 15 minutes. And then after they're done, we'll have nearly 30 minutes for your questions, which you can enter into the Q&A as Bailey instructed. And then to close things out, we'll have Claire Gelfman, the Foundation's very smart and impassioned Chief Scientific Officer, who will provide some concluding remarks. And while we'll be covering a lot of ground in the next 90 minutes, please remember that our website, fightingblindness.org, is a great resource for learning about research and clinical trials, our no-cost genetic testing program, which, by the way, has provided testing for more than 11,000 people, and our MyRetina Tracker patient registry, which has more than 22,000 registrants. So again, fightingblindness.org, it's a wonderful resource for our space. It is a promising time with more than 40 clinical trials underway for the inherited retinal diseases and dry AMD. And we have many more therapies, some of which you'll be learning about, moving toward human studies. Excitingly, the Foundation Fighting Blindness has provided funding for research that have made 88% of those trials possible. And I want to thank each and every one of you out there who supports our mission for helping drive the research, because ultimately it's you and the work you're doing in your communities that make the research possible. So without further ado, I would like to present or introduce our first speaker, and she is Jami Kern. Jami is an MBA and a PhD and Chief Clinical Officer at Nacuity Pharmaceuticals, whose lead program applies a gene-agnostic approach to treat RP, Usher syndrome, and potentially other conditions. And they have a trial for people with Usher syndrome underway in Australia. Prior to joining the Nacuity team, Dr. Kern spent 15 years leading clinical development and medical affairs at Alcon. She earned a Bachelor of Arts in Chemistry from Austin College, her PhD in biomedical science, biochemistry, and molecular biology from the UNT Health Science Center, and an MBA from TCU's Neely School. So, Jami, the virtual floor is yours.
Thank you so much, Ben, and thank you, FFB, for all your support, and for everyone for joining today on a Saturday for most of us, I think. Very pleased to be here and to be sharing an update from Nacuity. So as Ben mentioned, Nacuity is a clinical stage biotech company, and we're located in Fort Worth, Texas. Most of us have had our start or had a long career at Alcon. And we are focused primarily on developing a treatment to slow the progression of RP, retinitis pigmentosa. We have a small antioxidant drug. It's N-acetylcysteine amide, or NACA for short, that we believe will preserve vision by protecting cone photoreceptors from oxidative stress. And as most of you, I'm sure, understand, when you have RP, genetic mutations cause a loss of rod photoreceptors. And those are the cells that are responsible for low light vision and peripheral vision. And that's why often the first clinical signs are difficulty with night vision. As those rod cells die, though, there is also an increase in oxygen near the retinal tissues. And that oxidative stress is what then damages cone photoreceptors and compromises our central and our sharp vision and our acuity vision, which is really critical to how we function with our vision. So, as Ben mentioned, our approach is gene agnostic. We, rather than correcting the genetic defects in an effort to save the rod photoreceptors, NACA works to protect against the resulting loss of cones, and thus preserving our central vision. Our drug is a tablet. It's taken orally every day, and it's currently being studied, as he mentioned, in clinics in Australia to help us understand the safety of that chronic dosing, as well as to understand the potential for it to preserve cone function in patients with RP associated with Usher syndrome. That study actually began in 2020, and unfortunately, as we all know, that was a very challenging year for a lot of reasons. And unfortunately, for many of us, clinical research was no exception to that. However, even so, we have really seen great progress in the study over the last 2 years, and we're really pleased to be in the clinic and excited to see the progress. So in our study, patients are randomized to receive either active treatment or placebo, and they're followed for 2 years. And during the course of the study, we measure endpoints that are vision and also retinal structure related in order to understand if NACA is indeed preserving the cone photoreceptors. Because our treatment is gene independent, we actually have patients who have various types of Usher syndrome in the study, randomized or enrolled in the study. Those patients, so far we've enrolled 40 patients, and of those 40 patients, we have patients who have USH2A, we have patients who have USH1B, and also at least one patient who has USH1C. And we're continuing to collect that information just to understand the patient population, even though we don't expect it to play a significant role in the treatment efficacy. By the end of this year, we will have 36 patients who've been on active treatment, who've been on treatment for 9 months at least. Of those 36, 19 of them will have been on treatment for at least 18 months. So we really are starting to have a good amount of clinical data around the chronic dosing of the trial, not only for to understand the safety, but also to start getting to a point where we can potentially look at the efficacy. So far, we have seen very good tolerability and safety in the study. Patients have wholly been very compliant with their dosing. They've been taking their drugs mostly every day as they were instructed to do, missing very few doses. They've also been really incredibly great about attending their clinic visits as planned. And that's something that I am really thankful for. Our participants are amazing volunteers. This is a long study. It involves long, steady visits sometimes, and some patients are traveling very long distances in order to participate, and they've just been incredible. So we're really very blessed to have those participants. And I just want to say, for any of you who are thinking about engaging in a clinical study and participating in clinical studies, it's really a critical piece, and it's what makes us successful as we move forward in the development of our product programs. There's no way we could do it without you. So, you know, definitely consider participating if you have the opportunity to do that. So in fact, those patients who started the study at the beginning of 2020, when we started the study, they are rolling off of their 2-year planned study visits at this point, and they're beginning to end the study. But we had received indication from our clinical sites that the patients actually really wanted to stay on study treatment. And so we went ahead and worked with our clinics in order to amend the study protocol and allow patients the opportunity to continue on treatment after they finish the core study if they so desire. So that how that works, they finish their study, their 2-year study visit. They're given the opportunity to either stay on the study drug or their placebo, whichever they're randomized to, or they may complete the study as planned and move on with their lives. So far, every patient who's been on our study and who has finished has chosen to stay on treatment. That's really exciting to us for a couple of reasons. First, it tells me that the study drug is actually tolerable, and they're perfectly happy to continue to take an oral drug every day that might help them. But even more so, we very much hope that it also means that they feel like they're seeing a benefit, or at least some of them are seeing a benefit to taking the drug. It'll also give us a little bit longer longitudinal data as we move forward in the clinical research aspect, and it may really help us better understand how the drug works on a longer term basis, which is really useful to us. So we're very excited about the progress of the clinical study. Earlier this year, we conducted an interim analysis where we looked at the safety data from the study. We didn't unmask the data, so we don't know which participants have been randomized or assigned the placebo versus the active NACA treatment. But what we saw very clearly was that, at least to date, the NACA treatment has been safe and tolerable in these patients with chronic dosing, which is very exciting to see. We are continuing to enroll patients in Australia and plan to take our first look at efficacy data early next year. So by mid-year next year, we're hoping to have our first indication of how the treatment is working and potentially use that information to broaden and expand our clinical program. So with that, I think that's the big update from Nacuity. We're, again, very thankful for the support of all of you and Foundation Fighting Blindness, who certainly, without them, we wouldn't be where we are today, both from a scientific perspective as well as from a funding perspective. And we hope to have very positive results to share with you next year.
Thanks, Jami, for that great overview. And thanks for mentioning the foundation. We are funding Nacuity through our venture philanthropy fund, several $1 million. So this is an important project for us. And I do want to ask you a question now, because it's a question we've gotten from a few people.
Sure.
People out there seem to know about the NAC study going on at Hopkins. So you can talk,
Yes.
Can you talk about the difference between the two and the similarity, maybe?
Yeah, absolutely. It's a great question and really important to understand. So actually, our research and where we started is fully derived from the research that Peter Campachero and that group has been doing at Hopkins. Our drug is just the amide form of NAC, so it has an extra little addition onto the molecule. But the thing that that does. What that really does for us is it makes it more bioavailable and more active. So presumably we'll be able to take less of our drug and have a better effect. It also probably gets to the eye a little bit better. And Dr. Campachero, who's leading the NAC studies in the US at the moment, he did all of that preclinical work comparing NACA to NAC originally, and has worked with Nacuity very closely over the years, and we're good partners. We frankly just want to see something that works for patients. NAC was already approved as a drug, and so it was quicker to get into clinic in the US using NAC, and Peter did not want to wait, and we fully support that decision. And the data that he's generated from his NAC attack study and other protocols that he's run has really helped us understand the potential for our drug as well. So everything moves the science forward. My understanding is that at the point when we are able to provide NACA to patients in the US, Peter has said that he would likely move his program to NACA because it likely will be the more preferable version of the drug in order to not have to take massive doses for a lot of time. So, but both of them work in a similar way. They're both antioxidants. They both work in the same path. The amide version of the drug, which is what we have, just gets to the cells a little bit easier.
Right. Right. Thanks for clarifying the difference between NAC and NACA. And like you said, NACA looks like it may be the better option, but whatever works.
We don't know, but we will move forward. Yep, for sure.
Okay. So thanks, Jami. So next up, I would like to introduce Dr. Erin O'Neill. She's VP of Clinical Development at Opus Genetics, and they happen to be a company that we help launch, and we're also funding through our RD fund. And she is also a practicing ophthalmologist on the Inherited Retinal Degeneration's ophthalmic genetics team at Children's Hospital of Philadelphia. She completed her medical training at University of Pennsylvania and her ophthalmology residency and fellowship at Shea Eye Institute at University of Pennsylvania. She is a prior recipient of the Diana Davis Spencer Clinical Research Fellowship Award from the Foundation Fighting Blindness for her work to characterize the natural history of retinal structure and functional changes in people with choroideremia. So welcome, Erin. Great to have you.
Thank you so much. It's such a pleasure to spend part of my day with you all today. And thank you to Jami for that update. That was, as a clinician, very exciting to hear. I do have some slides. These are to keep me on track because I have a lot of ground to cover. So I'm going to share my screen. I will describe everything to you on these slides. But again, these are mostly just to make sure that I don't stray too far from what I want to share with you all today. So as Ben mentioned, I'm VP of Clinical Development at Opus Genetics. We are a unique gene therapy company that is focused exclusively on patients who have rare inherited retinal diseases. And I want to share with you a bit about our therapeutic pipeline today. First, I'm going to jump into a bit of background about our company, who we are, where we came from, and what our goals are on this space. So on this slide, I have a picture of Jean Bennett, who's one of our scientific founders, one of the masterminds behind Luxturna for the treatment of RPE65 retinal degeneration, as well as a board member at Opus. I'm also sharing some of the cornerstones of Opus Genetics and who we are. So the company was born out of discussions between Jean and our CEO, Ben Yerxa, former CEO of the FFB. And she had noted to him that there are a number of scientifically compelling therapies that have been developed and undergone preclinical testing for inherited retinal diseases, but which when taken individually can seem very risky for a company to take on just one at a time, given the molecular, meaning genetic diversity of these diseases and the deep nuances that differentiate one form from the other. But by grouping the therapies together and building a company dedicated solely to the understanding of the diseases and their treatment, that becomes really compelling from a corporate perspective. So we capitalize on the learnings from Luxturna, namely the dosing, route of administration, how to time treatment of the other eye, as well as any immunosuppression that needs to go alongside with treatment. And we also rely on de-risked methods of delivery of therapy. By that, I mean that we use AAV vectors exclusively, which have a long track record of demonstrated safety and efficacy when delivered to the eye, and we deliver to the subretinal space as is used in Luxturna. This is the basis for all of the programs that we have in our pipeline, which I'll be discussing today. We are committed to the highest quality manufacturing on a small scale that makes sense for ocular gene therapies. We also aim to build very strong relationships with clinicians and scientific experts in our field who can help us guide our clinical trial design to give our therapies the best chance of success. And most importantly, we never lose sight of the fact that we were literally built by patients, thanks to the partnerships and investments of the FFB and the RD Fund, and that ultimately we serve you. On this picture, I have photographs of the rest of our management team. In addition to Ben, who I also mentioned, we're joined by Ash, Joe, and Jennifer, who have decades of experience in the biopharmaceutical space. And then there's me, who usually is just happy to be part of the conversation and in the room. On this slide, I have photos of our scientific advisors. So we're joined by Jean Bennett and Eric Pierce from Harvard, who are our scientific founders, by Radha Ayagari at UC San Diego and Sanford Boy at University of Florida. On this slide, I have a picture of our clinical advisors. This board is chaired by Dan Chung, who was a key part of the SPARK team and development of the mobility test, which was pivotal in the approval of Luxturna. And he is joined by Dr. Heon of Sick Kids in Toronto, Jose Sahel at University of Pittsburgh Medical Center, and Bart Leroy, head of the department at Ghent University and one of my colleagues at Children's Hospital of Philadelphia. So then now that I've covered who we are, on this slide, I want to give you a bird's eye view of where we are at Opus and what we're working on. So here I have listed our first 3 pipeline programs, which are for LCA5, RDH12, and NMNAT1. Each of these 3 programs are for treatment of a form of inherited retinal disease known as Leber congenital amaurosis, or LCA, which is a very severe form of disease that is typically diagnosed clinically within the first year or 2 of life. Each of these genetic forms taken on its own is relatively rare, with an estimated prevalence of 1,000 or less patients in the United States. And each of them is also at a distinct phase in terms of its clinical development or readiness to be trialed in humans. And they're spaced out such that we anticipate being able to file 1 new IND or filing with the FDA to start a clinical trial in patients per year. But we won't stop there. I also note on this slide that we're exploring other therapies to potentially bring in-house to maintain this pace well beyond 2024. A bit more about LCA5 retinal degeneration. This is a very severe form of LCA, again diagnosed within the first year or 2 of life. Patients experience decreased vision, profound night blindness, and poor peripheral vision, as well as oftentimes nystagmus, or a difficulty with fixation due to their poor vision. What's very exciting from a therapeutic perspective, and what I'm trying to demonstrate in the photographs on the left-hand side of my slide, is that like RPE65 retinal degeneration, which again can be treated with Luxturna, or CEP290 LCA, for which there are many therapies under investigation, here we see a structural and functional dissociation of the retina in affected patients. That means that in these OCTs, or cross-sectional pictures of the retina of affected patients, we see excellent preservation of the necessary cells for visual function, often well into adulthood. But despite sitting there, they're not working properly. They're waiting essentially to be rescued with gene augmentation therapy. And we know from the success of Luxturna that this is an indicator that a gene augmentation approach is very likely to be successful. We know that the LCA5 gene encodes for lebercilin, which is displayed in the diagram I have on the right-hand side of the slide. Lebercilin is crucial in the transport of other proteins between the inner segment of the photoreceptors, which is the top part of this diagram, and the outer segment of the photoreceptors, the bottom part of this diagram. And these are the cells at the back of the eye that are crucial for sensing light and sending signals to the brain. We hope that gene augmentation therapy would restore the function of this protein and allow for normal transport within our photoreceptor cells. We know that mice with LCA5 retinal degeneration who are treated with therapy have both structural preservation and functional improvements in their vision. On the left-hand side of this slide, I have a variety of OCTs from mice who are affected by disease. And you can see shaded in the blue areas on their OCTs, the mice who are treated at 5 days postnatally and 15 days postnatally have relative preservation of their outer nuclear layer of their retina. And this corresponds with the 2 diagrams on the right, which are each measures of the visual function of these mice. So the chart in the middle shows the pupillary responses to light in treated mice. And so again, the mice who are treated at 5 or 15 days after life have significant improvements in their pupil responses to light. They also are able to perform significantly better on maze testing, which asks them to find a light at the end of a complex maze. They can perform this more reliably at 20-fold lower luminances after treatment. All of this holds a lot of promise for possible efficacy in patients. So our first in human trial will be a small one. And I have a photo of the study schema on this slide. We aim to enroll 6 participants, 4 adults and 2 children. Each participant would be enrolled sequentially, meaning we'll observe each patient for 30 days after treatment before we proceed to the next participant. We'll be exploring 2 doses in this study. So after the first 2 participants are treated at our starting dose, we'll pause and allow our independent data monitoring committee, which is made up of clinicians and experts in this field, to review the data and provide their recommendation to us as to whether to proceed to the next higher dose. We'll again pause after that higher dose cohort to allow that independent committee to make a recommendation on whether to proceed to enroll children in our pediatric cohort. All of the participants in this trial will receive the subretinal therapy. This is not a randomized trial. It'll take place at the University of Pennsylvania and Children's Hospital Philadelphia. And our primary objective will be to monitor for safety, but we'll also be studying a number of measures of visual function to try to elucidate any improvements in vision after therapy. This will include full field retinal sensitivity via FST testing, which I'm sure a number of the patients in this virtual room have participated in, measuring pupillary responses to light, and as well as performance on a novel mobility course, as well as patient-reported outcomes. Moving on to our next program, this is for RDH12, which is another form of LCA presenting within the first few years of life. This is more common than LCA5, affecting several thousand patients in the US and Europe. In this form of disease, we again see early vision loss and night blindness. On the left-hand side of this slide, I have 2 photographs of retinas from patients with RDH12. And here we see that patients have significant central structural changes, as well as retinal pigment changes from an early age. Interestingly, despite the profound appearance of the retinas, we actually see better preservation of rod photoreceptor function outside the macula, as well as cone-mediated foveal vision, that vision that we use to read the eye chart in clinic, in RDH12 patients, as compared to RPE65 patients with better retinal structure. That's what the chart on the right-hand side of this slide is getting at. The RDH12 patients are represented by the circles, and you can see that their ONL thickness, which is on the y-axis of this graph, is generally lower than the RPE65 patients who are the stars. And yet their retinal sensitivity numbers, which is along the x-axis, and here higher numbers is better, is actually higher than those patients. And so that provides an interesting window of opportunity for therapy. In terms of an update as to where we are with this program, we're performing ongoing preclinical and non-clinical studies to round out our understanding of the safety and efficacy of therapy. We've opened a dialogue with the FDA via a pre-IND meeting, which we completed earlier this year. And we've begun the process of manufacturing, writing the clinical protocol, and speaking with potential study sites both within and beyond the US for this study. We're also very excited to be a part of the RDH12 scientific workshop coming up this very next Monday with a number of clinicians, scientists, patients, and regulators to discuss the clinical endpoints which might be of the most relevant for this clinical trial design and these patients. Here again, I am showing our planned study design, this time for the RDH12 study. Again, our primary objective will be safety. And in this case, we anticipate 3 doses in adults before proceeding to a pediatric cohort. Each cohort will be slightly larger at 3 patients instead of 2. And again, all patients enrolled in this study would receive the subretinal therapy, not a randomized study. Again, we've built in pauses to allow our independent data monitoring committee to review all cohort data before proceeding. And we're very excited to share that we will be learning a lot more about the natural history of these patients and the possible endpoints through the UniRare study, which was shared by the FFB yesterday. And in my remaining time, I do want to also touch on our third therapeutic program, which is for NMNAT1 retinal disease. Like all of these programs, NMNAT1 is a severe disease which affects children within the first year of their life. It's estimated to affect thousands of patients in the United States and Europe. It's characterized by these central atrophic lesions of the retina, for which I have a picture on the left-hand slide, which have been observed in patients within the first few months after birth. Yet we know beyond those central lesions that there is preservation of retinal structure and function. NMNAT1 as a gene encodes for an enzyme critical in the synthesis of another enzyme called NAD. NAD is crucial in cellular metabolism, DNA repair, and immunity. On the right-hand side of this slide, I have the results from some mice who were treated with our proposed therapy for NMNAT1. They are represented by the blue bars in the blue tracing. So in the middle of the slide is a graph showing the preservation of retinal structure in mice who are treated with the therapy via subretinal injection, as compared to the untreated mice who are the pink bars. And then the ERG tracings, you can see that the improvements in the blue tracing, which is again the treated mice, approaches that of a wild type or unaffected mouse in terms of retinal function. As far as a clinical update for this program, we are performing additional non-clinical studies next year to understand better the efficacy of therapy. We have our manufacturing plans in place. We are planning for our pre-IND meeting with the FDA to inform our next steps and our trial design. And we're again planning for multiple study sites, which will be selected closer to the IND filing date. But we aren't going to stop there. As I said earlier, our goal is to maintain our pace of filing one new therapeutic program with the FDA per year to address many of the inherited retinal diseases that are awaiting treatment. This bubble chart on the right-hand side of my graph is really where we live at Opus, and we're trying to address each and every one of these with time. So thank you so much for the time today, and I want to thank the patients who have shared their photos and their stories with us. On this slide, I have pictures of Braden, Bella, Abigail, Macy and her mom, Jenna, Alan, and Kendall and her guide dog, Maya. Thank you so much.
Thank you, Erin, for that wonderful presentation. It's exciting to see Opus cranking out so many gene therapies. And one point of clarification, I know your first 3 gene therapies are targeting LCA, but you're not specifically an LCA therapy company. You will likely—
Great question. We are not specifically only looking at LCA genes. Those are what our first 3 programs target, but we absolutely are looking beyond that as well.
Right, right. Thank you. Thanks again.
Thank you.
Okay, to keep us on track, I'd like to introduce our last but definitely not least presenter, and that is Dr. Maria Csete. She is Chief Executive Officer and founder of OkuloVision, which is developing cell therapies for treatment of blinding diseases. She's also a board-certified anesthesiologist with subspecialty certification in critical care, and as a medical school professor led by a liver transplant anesthesiology team last at Emory University. Dr. Csete received her undergraduate degree from Princeton, MD from Columbia University, and PhD from the California Institute of Technology, where she studied the role of oxygen in stem cell fate. This work became critical to translation of several stem cell therapies, including OkuloVision's cone progenitor cells. So thanks, Marie. We're excited to hear about your cell-based therapy. And you can unmute.
Trying to unmute. You moved my picture around just as we were getting started. I really thank you for inviting me to this fantastic presentation with 3 women on the panel, which is really wonderful. I am not going to show slides, and I am going to take a higher level, less detailed view than the previous 2 presenters to tell you about some conclusions that we can make for cell therapies that have preceded ours and how we're going to shake up that field with the first cone progenitor therapy, which will be the first cone-based therapy in clinical trials as a cell therapy and the first truly regenerative cone therapy for inherited retinal diseases. So in the context, in that context of where we are in the cell therapy field generally, I'm going to first make some major points about what we can conclude from cell therapies in the eye. First, the current cell therapies for inherited retinal diseases have potential only to slow cone degeneration, and their improvement in vision will be by protecting vulnerable photoreceptors from dying off. We hope to change that by having a truly regenerative therapy. Second, there's no therapy in clinical trial that has shown efficient generation of cones, even from progenitors that should generate cones, that can generate cones in a dish. If a cell therapy is not truly regenerative, it won't be useful in the late stages of diseases. Third, cell therapy advances, we are finding out, are going to be dependent on the development of better delivery strategies for the subretinal space. And I will tell you about a partnership that we have developed with a device company to try to address the safety issues of getting cells into the subretinal space. And I will emphasize that the focus on retinal pigment epithelial cell transplantations has been a function of the lack of science to support cone-based therapies. So if you look at the ability to generate an eye cell from a pluripotent stem cell like human embryonic stem cells, it's relatively easy to generate retinal pigment epithelial cells to put in the eye to support the cone survival. But no one has been able to generate really mature cones that will function from embryonic stem cells and can be manufactured to clinical scale. We're going to change that as well. Most importantly, I think that these talks highlight that long-term optimal therapy of inherited retinal disease will require combinations of cell, gene, and small molecule therapies. And I hope that our cone progenitor cells will be a platform for incorporating these therapies down the road. So let me go into a little bit of detail on the work that has preceded us, because it is related to what we are planning to do. The first attempt at regenerative therapies for inherited retinal diseases was based on Reneuron's program, and it is back in action. So Reneuron has a mixed population of stem cells that have the capacity to generate rods and cones and other neurons in the eye. And they delivered them into the subretinal space using methods that we are learning from, and in RP patients. And the first 3 subjects did really quite well, again, using standard eye chart measures of vision. But after the dose was increased, they started to have some surgical complications. The study was halted for a while. And I think that is the major learning experience from their heroic entry into this field, that we need a better delivery system for control over the mechanical issues of getting into the subretinal space. We also learned from them that we need a frozen product to facilitate worldwide delivery. And we also learned about the need for limiting dose potentially. When the important thing is the good news, that when injected into the subretinal space, a stem cell population can indeed improve vision and that improvement can be sustained. Csete, as some of you probably know, is using a very similar cell population, but instead of addressing the subretinal space, they put their cells into the vitreous as kind of a depot site so that the cells can secrete protective factors for cones. However, by putting them in the vitreous, you won't have a regenerative process going on. So these cells, as has been shown in their clinical trials, will not be effective in late stages of disease when there's no cones to act on. But there's real advantages in their therapy. One of them is that the injections can be repeated, and Csete has completed a repeat injection trial in 30 subjects with retinitis pigmentosa. The Astellas clinical trial injecting retinal pigment epithelial cells into the subretinal space is worth commenting on. These cells were injected in suspension in the subretinal space, focusing on patients with Stargardt disease. The initial results were very encouraging, but the effects were not sustained enough for the company to continue. And what I took away from this clinical trial is that I worry that Stargardt's is a much more toxic environment because of the nature of the mutation in that disease than other cone dystrophies are. And so in our initial clinical approach, we're going to give our best shot to the cells working. So we will include patients with all inherited retinal diseases, likely except Stargardt's at the beginning, and then expand when we learn from the initial safety trial. So what have we got that's going to shake up this field and do all these things that I say we can do? We have cone progenitors. So they're not really a stem cell because they only make one thing. They make cones when you put them in the eye, and that's all they do. The advantage is that we have, I think, a more predictable outcome from our therapy and less variation about what a mixed population of stem cells will do in a variable host environment. The other real advantage, of course, is that cone dystrophies have been largely neglected in this space. And we think that we have the right cell to address cone dystrophies in particular. The technology changes that led to this breakthrough product were first, physiologic oxygen culturing. And we use levels of oxygen to expand our cells that Reneuron uses, Csete uses, and this is actually based on my Caltech PhD work. We're getting rid of some of the oxidant stressors that Jami talked about in our manufacturing processes that are toxic to the cells as you grow them, as well as in the disease process. We also learned how to basically pan for gold. So we use a series of filtering experiments using flow cytometry to pull out cells from this mixed stem cell population that are destined to become photoreceptors, grow those up, and then use another panning kind of procedure to pull out cells that are destined to become cones. This serial selection process yields cells that are in what I call a developmental sweet spot. That's very unusual for stem cell therapies. The cells are still highly proliferative at the time of transplantation, but they're far enough down the path to becoming cones that they choose only to become cones after they're injected into the retina. So the proliferation gives us an advantage in scaling up our manufacture, and the differentiation gives us first a mature cone after transplantation, but also more predictable outcomes. Our preclinical studies show that we also have much more efficient generation of cones and integration of cones into the retina of any cell therapy that has been studied in animals before. The integration may be as high as 85% in the retina. And that is just off scale for other cell therapies in the eye, or really for that matter, any other cell therapy in other organ systems. Furthermore, much to the delight of our scientists, the cells integrate into a normal cone anatomic pattern, a mosaic pattern. And some of the clumping that has been seen with other cell therapies injected into the retina is not seen. This efficient integration into the retina translates into improved visual function by several measures, by improved electroretinography, and in particular the B wave, suggesting there's functional integration into the circuitry, and by improved tracking with the optokinetic system. So what will it take to get this into the clinic? Our company was founded in February of this year, and we still have a lot to do. In particular, we have to do efficacy studies with cells that are generated in a GMP facility. So we've chosen to go to GMP manufacturing sooner than the other companies so that the same cells can be used in the animal efficacy studies that we use in the patients. And we've developed a wonderful partnership with a company called STEM Matters in Portugal to develop our manufacturing protocols. And that's why I'm sitting here in Portugal, having worked with them this week. We also have decided to use the new immunocompromised RCS rat model developed at USC and Mark Humayun's lab for these efficacy studies, so that we won't have to worry about the toxicity of the immunosuppressant drugs in these trials. We're also planning dose-finding studies in non-human primates because we have no cell therapy from which we can choose a rational dose. Our level of integration efficiency is so high compared to, say, the reneuron cells that basing it on their experience makes no sense at all. But we're going to use the opportunity in these dose-finding studies to work with a company called AltaViz on a completely new delivery strategy for getting cells into the subretinal space. It's a largely computer-driven strategy so that the mechanical damage from a surgeon's thumb is really minimized, but the surgeons love this because they can visualize the cells going into the eye in real time during the injection. We think this is a major safety advance for getting cells into the subretinal space. We've engaged investigators from the world-class Doheny Eye Institute to lead the clinical trial, and in particular, to really deeply image these patients using Doheny's unique imaging capacity. Our clinical trial will also be a little bit unusual. We've decided that we're going to treat phenotype rather than genotype. So all inherited retinal diseases with central vision loss will be eligible for study. And we will base our inclusion criteria on making sure that in the first trial, just for the first patients, that the rest of the retinal circuitry is relatively intact so that we will give the cells the best chance of working. And I have to say that when I started talking about this, there was puzzlement because we are so focused on the different mutations that drive these diseases and making specific therapies for them. But I think that this approach will serve us really well in putting the cone progenitor cells forward as a platform for a broad range of diseases, first the inherited retinal diseases, and then moving on to macular degeneration. And I should say that I'll take the opportunity to announce that Paul Sieving, the former director of the National Eye Institute, has signed on to our science advisory board. And I'm very excited to be picking his brain about the real nitty-gritty details of our clinical trial design. So those are the major hurdles we have to go through in getting to the clinic, but we have world-class partners lined up with STEM Matters for manufacturing, Altavis for device and delivery, and the Doheny Eye Institute to lead the clinical trial. And our major hurdle right now is to take advantage of these fantastic partnerships to raise money in what is a difficult economic environment. But we feel that we have a very commercially viable cell therapy as a single dose for a sustained therapy that's gene agnostic and ultimately will be important for inherited retinal diseases and in AMD in combination with other cell therapies. We're confident that we can be effective in actually potently reversing central vision loss in patients with IRDs, and combined with other therapies, particularly retinal pigment epithelial cells, we can address the major unmet need of central vision loss from age-related macular degeneration. So our website went live just a few weeks ago, www.okulovision.com. Okulo is the Esperanto word for eye. And I invite potential partners and investors certainly to be in touch with us through the website and follow our progress there as we hope to get to the clinic as quickly as we possibly can.
Great. Thank you, Marie, for that great overview of your emerging cell-based therapy. One comment I wanted to make that I think you illustrated well, and that's that you've learned a lot from other trials that have not led to therapies, for example, Reneurons, and you've learned from Csete as well. And I think that's an important point for our audience, is that research, even if a therapy doesn't cross the finish line in a clinical trial, there's knowledge gained, and we learn from it and move forward. So I want
to make one correction though there, Ben, because Reneurone has resumed its clinical trial. So although they had a pause, they started again. I looked on the website because the Science Advisory Board, which I served on, has not met for several years. But the study was restarted a year ago in Oxford.
For retina?
For RP.
Really?
Yes.
Okay.
I was surprised to see it too, but I'm really glad to see it. I think that, the study was halted not because the cells were a problem, but the delivery was a problem. So it makes sense that they've worked their way through that.
Okay. Another point I wanted to make, I realized, Marie, OkuloVision is not targeting Stargardt disease. That does present some challenging, there are challenging aspects to that, but there are other companies that are pursuing cell-based therapies for Stargardt disease. So I just wanted to make that point.
So we're not saying that we're not going to target Stargardts, but we think like macular degeneration, we need to have our cells go in there armored with RPE. So that'll be a combination therapy down the road. Right.
And that's an important point. You need not only photoreceptors, you need that supportive layer of RPE. Okay. We'll go to Q&A. I'm going to ask Claire Gelfman to join us for that. But we have received a lot of questions on NACA. And the two big questions is, will NACA move into a trial in the US? And obviously the first trial is for people with Usher syndrome, but what other conditions do you think NACA might be applicable to?
Yeah, those are great questions. So first off, why USH? And I think really our focus is on RP as a whole. But when you do a clinical study, you sort of have to balance looking at a broad population with focusing the population enough so that there's not so much variability so that you can actually see an effect if there is an effect, a treatment effect. So we were hoping that the initial USH study would serve as a clinical proof of concept and give us the opportunity to show in a relatively small sample size that the drug is actually working for that purpose. So that's why USH right now, but really anyone with RP theoretically should be benefiting by the approach. And in fact, there's potential for any disease that has an oxidative stress component to have a benefit of using that, depending on the progression of the progressive state of the disease. We have not looked at all of those options and all of those opportunities yet, but we certainly have had conversations internally about other opportunities, and assuming that the study is successful in the RP world, then we'll probably expand into some other potential oxidative stress diseases. Regarding doing the study in the US, it was quicker for us to go to Australia. That's why we're there. Getting the trial started in the clinic is really critical to helping us understand what's going on. It was quicker, and frankly, it also is a little bit less expensive resource-wise for us to do that. So that's why it's in Australia right now. We do have an open IND here in the US that is a clinical stage IND. We are amending that IND right now. So within the next couple of weeks, we'll be submitting that amendment with the safety data from the clinical study in Australia. And also with a protocol that hopefully will allow us to do this kind of study in patients in the US. Depending upon what happens when we look at the efficacy data early next year, we will make decisions about what the next step really should look like. So it may be that we see very positive results in the initial poll of the data for the USH trial. I certainly hope that that is the case. If it is the case, it will allow us to move more quickly forward with the program. If we see an indication of something working in the right way, but just that we clearly need more time, we might have to put a pause and watch the patients over a longer period of time, or potentially try to enroll additional patients in order to bump up our sample size to really understand what's happening. Our goal is to move the program as quickly as possible, but we certainly, want patients to be doing something that's safe and that might actually be effective for them. So that's sort of where we're at the moment.
Certainly. And it's always a little more challenging to measure and observe an effect in a treatment that's designed to slow progression.
Yeah.
And obviously it can be a very valuable therapy, because slowing progression can be a wonderful thing for people who still have some vision, but people don't get that big boost of vision that's very obvious early on. So thanks for all that information. I know a lot of people are excited about the work you're doing. So the next question, it's more of a general question, and I want to ask it of both Erin and Claire, because I think there are a couple ways to answer this question. But it came in from somebody, I think, who has advanced RP, and they're wondering if there's a therapy for them if they've lost a lot of vision, let's say, because it's allegedly, they've potentially lost a lot of rods and cones. But I guess the point I want to have you re-emphasize, Erin, is there are some diseases where people have lost a lot of vision, but there are a lot of photoreceptors remaining. And can you expand on that? Because the point there is somebody could have, they could be nearly blind, but still have an opportunity for gene augmentation.
Yeah, absolutely. So what gene augmentation therapy strives to do is allow the cells to have a restoration of their naturally occurring function. So typically the pathway, the way that I think about it in my head, is if you have a disease in which the cells are not able to function due to whatever reason, a transport protein isn't working, an enzyme isn't working, that allows the visual cycle to progress. Those cells will not sense the light and send the signals to your brain for interpretation. They will slowly start to become more and more senescent. Over time, due to pathways that I don't fully understand, to be perfectly honest, that lack of function, kind of use it or lose it, lets the cells die off, and they start to lose their support network. The photoreceptors start to lose the RPE that supports them. And so gene augmentation therapy strives to rescue those cells that are not functioning properly, but are still there. It can't restore cells that have been lost, but there is an opportunity to improve vision as long as the cells are still there. And we see that time and again with our patients with Luxturna. They have poor function. They don't, you can't see your own retina. They don't know exactly what structurally is there. We can see it when we do our OCTs in the clinic, and it gives us a clue that they're likely to have improvement in the function. It's important to have both of those pieces understood on an individual basis. What structure is there and what level of function is there?
Right. Thanks for emphasizing that point. So you have to know what the retinal structure is to know potentially what approach may be helpful. So part 2 of that question, I'd like to direct to Claire. For the folks out there that maybe have advanced RP or advanced Stargardt disease, where they've lost all their rods and cones or most of their rods and cones, what may be beneficial for them?
Yeah, thank you, Ben. This is Claire Gelfman. So there are clinical trials in progress that are all about restoring vision, as you said, to those who have lost a lot of their photoreceptors. As Erin just beautifully described, for a gene therapy approach to work, you have to have a cell that's there to receive the therapy. If the cells have died off and they're gone, there's nothing there to receive it. So we talk a lot about a type of therapeutic approach called optogenetics. And the word frightens a lot of people away, but I'm here to say that it's actually a simple concept. And that is that functional vision is there because when light comes through your pupil and is received, if you will, from your photoreceptors, those outer segments of your photoreceptors, a message is transported to your brain so that you can interpret the image in front of you. So that ability of the photoreceptors to capture light is really important for proper vision. But if you don't have photoreceptors to capture that light, then your brain never has an opportunity to interpret what's in front of us. So what optogenetics is all about for those late stage retinitis pigmentosa patients who don't have functional photoreceptors is an ability to give that ability to respond to light to a different part of the retina that's not degenerating. So the retina is actually made up of a lot of different layers. We've talked a little bit today about the retinal pigmented epithelium, the single layer of cells just beyond the retina that's important in different types of macular degeneration. But we haven't really talked about the intricacy of the layers of the retina. We've got the rods and the cones, and there's different layers, and there's actually an inner layer called the ganglion cells that is really what feeds messages to our brain through our optic nerve. And what's interesting is that you can have almost no vision, very few photoreceptors, if any, but your ganglion cells are still present. So what optogenetics is all about is giving that ability to respond to light to the ganglion cells. And in fact, that strategy has been tested, that optogenetic strategy has been tested in human clinical trials. A few different companies, Gesite is in phase 2. Actually, some very interesting results have already been reported where it's a restoration of vision, not just no further damage, where individuals with little to no light perception were able to make out objects on a desk and see a crosswalk in a street. So this is not, reading fine print of the New York Times, but it is a vision improvement that means so much to someone who just didn't have it to start. And the cool thing about optogenetics is that one company, we were saying before about the learnings from other companies that are evaluating different types of therapies. This is another great example where there are a lot of companies that are tweaking the optogenetic therapy to try to do better, to try to help those restore vision with later stage photoreceptor degeneration. Gesite is one of those that I mentioned. There's a company called Kiora that is also using an optogenetic therapy. There's a company called Videri Bio. And, I just want to bring all those names up just to let you know that we're not banking on one single company bringing this forward. There are a lot of people who are, with each trial, we're learning from our colleagues to do it better and better. So there will be a treatment for those who have later stage photoreceptor degeneration.
Thanks, Claire. Yes, optogenetics is very exciting with so many options in the clinic. If we can get one or even two of those across the finish line, that would be awesome.
And probably worth mentioning the possibility of retinal implants down the road too.
Absolutely, absolutely. Retinal implants, cell-based therapies, these agnostic approaches, it's that toolbox approach, kind of multi-pronged, lots of shots on goal. So we're all working together for the common goal of moving treatments to the clinic and to approval.
Right. Thank you. Actually, I want to address a question about genetic testing, and just to clarify our programs a bit. So the Foundation Fighting Blindness for now more than 5 years has been offering no-cost genetic testing to anybody in the US. It is only for people in the US that are clinically diagnosed with an inherited retinal disease. And it's the doctor who orders the test. You don't order the test. Hopefully your doctor will be amenable to doing that. And then the doctor takes, usually it's a saliva sample or a cheek swab, sends it out. The results come back in about 4 weeks. You get free genetic counseling as part of that to help you understand the results. We offer that through Informed DNA, or the doctor can offer it through his or her clinic. Now, the registry, the MyRetina Tracker registry, is a global patient registry. Anybody, whether they've had genetic testing through us or not, can use the registry and enter their information so that they can get on the radar screen of researchers and doctors that are doing clinical studies. And so one of the questions was about, do we share data on patients with the research community? And the answer is yes, we share a lot of data. Now, we're not sharing your personal information. This is all de-identified data. I think that's important to understand. But we're sharing information on genetics, the phenotypes, as we like to say, how the diseases are affecting the retina, so they can better understand the conditions and potential therapeutic targets. But again, the MyRetina Tracker registry is global, and we encourage everybody with an IRD, whether they know their gene or not, to get into the registry. So thank you for giving me a moment to talk about that. One question we had, Erin, I wanted to direct to you about your RDH12 gene therapy. Is that for really all forms of RDH12, whether somebody has LCA or RP?
It's a great question. I can't say that we've made a final decision as to the writing of the protocol, but when you think scientifically about it, the gene augmentation therapy should work no matter the clinical diagnosis, as long as the genotype is correct.
Right. So thanks for clarifying that. So stay tuned. And hopefully, if somebody still has enough photoreceptors left, they'll be eligible. So I want to ask you another question, Erin, and this is related to what somebody asked about the FDA. So some of the therapies that we've been talking about are designed to slow vision loss or halt vision loss. And the question is the FDA amenable to therapies that slow vision loss as opposed to restoring vision? And I want to broaden that question to what is the FDA ultimately looking for if they're going to approve a potential therapy in a clinical trial?
Yeah, so to answer the last part of the question first, their responsibility to the community, which they do a great job of, is only allowing therapies that have a clinically meaningful and safe effect on patients. And so their ultimate goal is to understand what is the exact effect of this therapy and is it meaningful to patients? Is the prospect of the direct benefit to an individual patient greater than any possible associated risks of the therapy itself? And so to get into the nitty-gritty of it on an individual program basis, you'd have to ask them individually, you know, is this enough? What we're demonstrating, is this enough or is it not? The very interesting part to me about your initial part of your question is the slowing of progression is hard to define, especially in a rare disease in which the acquisition of natural history data is a challenge. And so I just want to use this opportunity to plug that if anyone has the opportunity to participate in a natural history study, you have no idea how helpful that is to us in our clinical trial design to be able to say, we know that patients with this gene should progress structurally or functionally at a given rate per year. And when they receive the therapy, that doesn't happen. The progression is less. And that, I think, is something that would be really meaningful to the regulatory agencies.
Right. Great answer. Great
Pick up with that a little bit too, because in the macular degeneration space, the OP-REGEN lineage trial is a rate of slowing of the accumulation of area of atrophy as their endpoint. So the FDA does allow those kinds of therapies to go through, endpoints to go through.
Right. Great point, Marie. And that sort of leads to a question and a really hot topic about endpoints. And Claire, maybe you can take a moment before your final remarks, or you could really lead into your final remarks, depending on how you want to handle this, about clinical trial endpoints and what the foundation is trying to do, or we're not trying, we're really funding a lot of research to come up with better endpoints.
Yeah, thanks, Ben. This is Claire Gelfman again. Certainly, when we think about successful clinical trial, we have to, at the outset, before a single subject receives the potential medication, we have to define how much of the drug is being given, how often it's being given, and how we are going to define success in that clinical trial. And the definition of success really comes from the choice of an endpoint. Will it be, for example, phase 1 is typically about safety. You know, the FDA is all about safety. In a phase 1 trial, when you think about areas outside of ophthalmology, you might notice that phase 1 is all healthy individuals without disease you ultimately want to treat, because it's just a simple question, is the drug safe? Now, we can't do that for ophthalmology studies because most, but not all, require delivery of the drug directly into the eye. And obviously, we're not going to take healthy individuals. That just would not be ethical to do that. So that being said, that the safety parameters have to be decided upon before the trial starts to find success of a phase 1 study. Phase 2, the secondary endpoints of a phase 1 trial, and then further phase 2, phase 3, now we're really getting into not only safety, but efficacy. Is it actually working to restore vision, delay further progression? So how do we do that? Is it an eye chart? When you go to an ophthalmologist's office and you're reading those letters, I mean, you can have tunnel vision due to retinitis pigmentosa and read that eye chart beautifully. So how effective then is that really as an endpoint? So the choice of an endpoint is really critical. You know, with Luxturna, the only approved gene therapy for an inherited retinal disease for individuals with mutations in the RPE65 gene, this question came up. How are we going to assess that this drug is working? And so what Spark did, the company that developed Luxturna, they went to the FDA and they said, we have an idea that we think can really tell us whether or not meaningful vision has been improved. And it's a human maze. So I'm going to say that again. It's a human maze. It's exactly what you think. And so these individuals, essentially, you know, before and after, when they first went through it, they were bumping into the walls, and afterwards they weren't. It was incredible. And then one step further, through conversations with the FDA, that human maze was performed under different light intensities, you know, low light versus, you know, darker conditions or lighter conditions, to really assess the efficacy of the drug. And all those discussions were had with the FDA before the trial actually started. And so what we are doing as an organization to support the patient community for all these trials is to help come up with endpoints that the FDA will agree to in terms of establishing whether or not a drug is truly helping patients not just look at lines on an eye chart, but, you know, lead a more meaningful life. So you might ask, how do we find those? Well, Erin mentioned natural history studies. You know, those are so important because when you can, a natural history is essentially following an individual through a period of time, looking at how the course of their vision loss, but also how their lives are impacted. So it's not just, you know, I can't see off to the side as well, but driving, looking at a computer, being able to read a newspaper. You know, everybody has kind of their own thought of what functional vision is. And so we use natural history studies that we perform through the foundation. We enroll subjects for these natural history studies through our registry. And in fact, a very unique natural history study is there was a press release that came out just a day or two ago about a way to look at the natural history of some of these very, very rare inherited retinal diseases. So the name of the study is UniRare, UNIRARE. And we are able to follow the course of the disease in 1,500 patients that are going to be enrolled. And the second part of this is to focus on specific patient populations over time, so that we can then learn about meaningful endpoints and then communicate that to the FDA. And a lot of times, those are again not just some of the more intricate assessments, imaging techniques that might happen from your ophthalmologist, but also quality of life questionnaires that are given, you know, and this is where we really learn about meaningful vision improvements. And then our job is to then go to the FDA and report this information so that we can help identify what those endpoints might be. And in the past several years, there are endpoints that are now agreed upon by the FDA that came out of natural history studies that the Foundation Fighting Blindness actually funded. So it's a big area. We have actually next summer an FDA endpoints meeting where we are going to continue this effort. We had one specific for X-linked RP several months back, and you know our goal is to capture as much information from individuals who are living with these inherited retinal diseases, take that information and then suggest what those endpoints might be, but most importantly, collect data to support those as endpoints so that when we go to the regulatory authorities, you know, we're not just suggesting it, but we have data to support them.
Thank you, Claire. So we have a few minutes left before we conclude. Claire, did you have anything further you wanted to add for your official closing remarks? I sort of led you down the path.
Yeah, you know, I'm just, you know, it's been such an amazing, you know, hour and a half here. And I'm looking, you know, on my computer screen, having just heard from the rock stars in our field, truly, who, you know, I mean, I speak to individuals who have inherited retinal diseases and dry AMD, another large focus of the foundation. And it gives me great hope to see all the different types of potential therapies that people are spending their life's work on. Today we heard about some gene-specific therapies from Erin and how those are already, soon to be in the clinic, which is so exciting. These are areas, individuals with mutations in the genes that you mentioned, Erin, there was no hope in terms of gene-specific therapies before Opus took these on. And I am so happy that, there's been such quick movement of development of those programs. And I look forward to focusing in on that last slide of yours with all those different inherited retinal diseases. So the approach that you and Opus is taking can be applied to many more than just those three, And then Jami, your agnostic approach that's really capitalizing on a pathway that is found in basically every patient with retinitis pigmentosa. We know that the retina is the most oxidatively active organ in the body, and too much oxygen is just not good. And in fact, if you already have, a genetic predisposition, oxidative stress can really cause major photoreceptor degeneration. And we know that from the multitude of preclinical trials and animal models of retinitis pigmentosa, whether it's from Usher's or no matter what your causative gene is, you may not even know your causative gene, but you've been told based on your lack of peripheral vision that you probably have RP. You hopefully can get free genetic testing from the Foundation Fighting Blindness. But if not a genetic-based therapy, it's so great to know that there is a single treatment, this NACA that you beautifully described, that could be given as a pill daily to help patients who are suffering from all different types of photoreceptor degeneration, because preserving that cone function, that's going to be such a meaningful increase in vision. And so, the gene-specific and then the more agnostic approaches, I think really cover so much of the people we want to treat. And then Marie, the cell-based therapies that you beautifully described, that area, there's so many opportunities there to restore cellular function, whether it's using a progenitor cell like you described that can be delivered with an ability to still divide to make more once it's back in the retina, but that it's programmed to become a functional cone and give us back the ability to see central vision, high acuity vision, color perception. I mean, that also incredibly meaningful. So, Ben, I'm sure it was deliberate and calling on these three rock stars in our field, but because it really covers so many types of therapeutic approaches that I hope can be applied to everyone on this call, and with the learnings from other companies, with different types of gene therapies and different types of cell-based therapies and different types of agnostic approaches, we learn about best ways to deliver the drugs and best ways to assign, success of a clinical trial. These are all approaches that the Foundation Fighting Blindness is supporting, in that we want to help our patients, find the best therapy for their specific situation. And so, as you can imagine, so much of our outreach as well is to ophthalmologists and scientists across the world to support all of those treatments that are being developed. And, I can't thank all of you enough, those who are on the call today as attendees. Also, I want to thank our incredible speakers and all the work that you and your respective companies are doing to help the Foundation Fighting Blindness achieve our mission, which is to eradicate these different inherited retinal diseases, as well as macular degeneration, the dry form, as right now unmet medical needs. But I'm hoping that, there's a logo for cancer, making cancer history, and it's sort of a scratch out. I'd like to see that for inherited retinal diseases. And I think we can do that with the amazing work that's happening by those that we heard about today and many others too. So thank you again for your attendance, participation, and all of the questions that we received. If we did not get to answer them, we will absolutely be contacting you to provide answers and an open dialogue should you have additional questions or comments. So thanks again, everyone. Happy Saturday.
Thanks, Claire. And thanks to our panelists. Wonderful presentations. Thanks for taking time out of your Saturday. And thanks to all of you out there in the virtual world for participating. You weren't virtual. You were really with us. So thank you.
Thank you.
Bye-bye.